On to the second main stage of cell cycle is mitosis. Mitosis is a form of nuclear division, and it only occurs in somatic cells. Mitosis can be divided into 4 steps. The first being prophase. Prophase is the longest phase in mitosis. Hence why if you were doing lab work on mitosis and you're viewing dividing cells under a microscope, you will probably notice that most of the cells will be in the prophase stage, as it is the longest phase of mitosis. And there will be a higher chance of you catching it in the prophase stage when you observe the dividing cells. In early prophase, the DNA is still in a form of chromatin. This chromatin will coil and condense to form chromosomes. In the early prophase stage, the nuclear envelope and the nucleus is still present, hence why you can see it here. In late prophase, the nuclear envelope disintegrates and the centrosomes will move to opposite poles of the cell. When it's at the opposite poles, it will start to form microtubules, which will then be used to form spinofibers. These spinofibers will then attach itself to the centromere of the chromosome, as you can see here. Now we're in the metaphase stage. Do you notice the difference between this diagram and the diagram above here? Yes, the chromosomes are aligned at the equator of the cell, which is this blue line right here. So as you can see, the spinofibers have aligned the chromosomes to the equator of the cells. And at this point, the sister chromatids are still attached by the centromere. Now take a look at this other diagram of a plant cell. Notice the difference between the spinofibers. Now let's compare the diagram of the animal cell in metaphase with a plant cell in metaphase. As you can see, the chromosomes are still aligned at the equator of the cell. However, there is an absence of centrioles in the plant cell. This causes the spinofibers that have been formed to not being able to focus on the centriole. Hence, the spinofibers will be straight and parallel to each other. Now let's move to our second last stage, the anaphase. This is a very rapid stage in which the spinofibers will shorten, causing the centromeres to divide and separate the sister chromatids. The spinofibers will then pull these sister chromatids to the opposite poles of the cell. The final stage of mitosis is telophase. So here, the chromatids have already reached the poles and become distinct, and are now referred to as chromosomes. These chromosomes will then unwind into its longer chromatin, and the nuclear envelope and nucleolus that had been disintegrated in the prophase stage will be formed around each group of chromatin, therefore forming 2 nucleus. And lastly, the spinofibers will then break down and disintegrate. Okay guys, we're in our last stage of the cell cycle, which is cytokinesis. So cytokinesis is the physical process of cell division, which divides the cytoplasm of a parental cell into 2 daughter cells. In the animal cell, the microfilaments, which are proteins, will attach to the insides of the cell membrane at the center of the cell. These microfilaments will then contract and pull the membrane inwards, creating what is called a cleavage furrow. The microfilaments will contract so hard that the parental cells will then ultimately divide into 2 genetically identical daughter cells. So it is very important to know that only animal cells are capable of cytokinesis. So you might be wondering, well, if cytokinesis can only happen in animal cells, then how does plant cells form its 2 genetically identical daughter cells? Well, in plant cells, there will be vesicles containing cellulose that will align itself to the equator of the cell. And these vesicles will then form segments of the cell wall to form a cell end plate. These segments will fuse together to form a new cell wall that separates the cell. which results in 2 genetically identical daughter cells forming. And that is the end of mitosis. So thank you guys as always for watching this video, and please do check out the other videos that we have on our platform, and I'll see you guys again next time. Bye!